Alrighty, so my name is Daniel Chee, and I'm a graduate student at the University of Washington, working with Professor John Stamatoyanopoulos. And today I'm going to tell you guys about a project I've been working on that looks at the interactions between microRNA and transcription factors. And so every cell in the human body is composed of the same genome. But however, when stem cells divide and differentiate, they undergo changes that result in vastly different phenotypes. But since these different cells and tissues are composed of the same, of a common genetic blueprint, these changes, these differences in phenotype have to be due to epigenetic changes. And since epigenetic changes are sort of defined by differential gene expression, and gene expression starts at the level of transcription. It's transcription factor DNA interactions that anchor these changes in the epigenetic landscape. And so in our lab, we leverage the fact that DNA is organized into chromatin, and in order for genes to be expressed, the chromatin has to unwind, making these actively regulated regions susceptible to nucleus digestion. So by treating a cell with DNase-1 and sequencing the resulting fragments, you can get an genome-wide snapshot of the areas of the genome that are being actively regulated. And so here I'm showing a shot from our genome browser, and so in this image, these peaks here correspond to areas of the genome that are sensitive to DNase-1, the so-called DNase-1 hypersensitive sites. And one thing that you can see from this image is that if you look at the epigenetic landscape across multiple cell types, there's areas of the genome that are sensitive to DNase-1 across all cell types, and there's also areas that are sensitive to DNase-1 in only a single or a few cell types. So this idea of DNase-1 hypersensitivity has a cell-selective aspect to it. And so if you were to actually focus in on one of these DNase-1 hypersensitive sites, you'll notice that the nucleotide cleavage isn't uniform, but instead there's actually areas of the genome that are protected from nuclease digestion because they're bound by a transcription factor. And the transcription factor that's binding a given DNase-1 footprint, as they're called, can be identified by looking in the footprint for the recognition sequence of known transcription factors. And so this idea of a DNase-1 footprint it corresponds to ChIP-seq data for these known transcription factors, and also phylogenetic conservation within these DNase-1 footprints. And so using this idea of a DNase-1 footprint, we can construct regulatory networks by first looking for DNase-1 footprints in the promoter regions of genes, and then next identifying other areas of the genome that are targeted by this transcription factor with a known regulatory sequence. And so by looking for DNase-1 footprints in the promoter regions of genes, we can construct this regulatory network, this transcription factor regulatory network. However, transcription factors, though they're a major class of regulating factors, they're not the only. They don't operate in isolation. Instead, other factors such as microRNAs are also important in regulating transcript abundance. And so microRNAs are short, roughly 21 base pair RNAs encoded in nuclear DNA. And once they're processed by Drosha and Dicer, they're incorporated into the RISC complex, where they silence translation of genes in a sequence-specific fashion. And so since this is done by Watson and Crick base pairing in what's known as the seed region of the microRNA, and it's believed that somewhere between 30 and 60% of all human protein coding genes are targets of microRNA. And so another significant thing is that many microRNAs regulate transcription factor, actually target transcription factor encoding genes. And so by using the same idea that I introduced earlier, we can incorporate microRNAs into transcription factor regulatory networks by searching the promoter region of microRNAs for transcription factor for DNase I footprints, and also by using microRNA target predictions, we can get this transcription factor microRNA co-regulatory network. And so this is sort of an overview of the pipeline that we used for this process. And so we use DNase I footprints from 102 very diverse, 102 cell types from diverse tissues, that are also from diverse developmental time. And so we identified footprints that contained known transcription factor binding motifs, and we combined them with target scan microRNA target predictions. And so this resulted in roughly 6,000 to 8,000 unique transcription factor microRNA interactions. And so one of the things that we noticed upon applying this method to our data is that we noticed that transcription factor microRNA feedback loops were abundant within this data. And so transcription factor microRNA loop is just basically when a transcription factor is present in the promoter region of a microRNA, and that microRNA targets that same transcription factor. And so across the 102 cell types, we found basically that the number of feedback loops across the cell types ranged from 33 to 172. And these transcription factor microRNA feedback loops were roughly cell-specific because only 11 of them were common to all cell types. And another thing we noticed is that these transcription factor microRNA feedback loops don't occur uniformly across all transcription factors. Instead, there's actually a set of transcription factors that are responsible, that are participating in these feedback loops within all of these interactions. So in this plot, I have transcription factors on the x-axis, and on the y-axis, I have the proportion of all regulatory cycles that a given transcription factor makes up. And you can see from this plot that SP1 and PLAG1, which are the two highest participants, make up roughly 50% of all regulatory cycles across all cell types. And likewise, if you look per cell type, you can see that there's certain transcription factors that participate in feedback loops in every cell type. So what I wanted to see was what distinguishes these transcription factors from those that don't participate in feedback loops. And so the first thing I wanted to do was look at their position in the network architecture. In their position in the transcription factor networks. And so the first thing I did was I looked at a measurement called the normalized network degree. And so here's the formal definition of the normalized network degree. But the very simplistic way that I like to think about it is the normalized network degree is a measurement of, if you were to remove that node from the network, how many of the total edges would you be removing from the network? And so in this, for instance, if you were to remove transcription factor 3, you'd be removing 5 edges and roughly 40% of the network of this hypothetical network. Whereas if you were to remove transcription factor 10, you would be removing 9 edges and roughly 75% of the network. And so in this case, transcription factor A would have a higher participation in the network and therefore a higher normalized network degree. And so I looked at the normalized network degree of transcription factors that participated in feedback loops versus those that didn't. And so in this plot on the x-axis, again, I have cell types. And on the y-axis, I have the median normalized network degree. And the magenta points correspond to the median normalized network degree of transcription factors that participated in feedback loops. And the cyan points correspond to the median normalized network degree of transcription factors that don't participate in feedback loops. And you can see that the transcription factors that participate in these feedback loops have a higher median network degree. And this plot just shows a distribution of the p-values that I got from using the Kolegmarov-Smirnov test. And it just basically shows that across all these cell types, this group was significantly higher. And so just because a given transcription factor has a high normalized network degree, that doesn't really say necessarily whether or not that transcription factor is driving the expression of the genes that it's connected to. For instance, if you take the canonical feed forward loop, all three of these transcription factors have the same normalized network degree in this case, but you can see that one of the transcription factors is actually driving the expression of the other two transcription factors, and where the other transcription factors are just sort of passengers in this entire process. So I wanted to look at whether transcription factors that participated in feedback loops were also in the driver position in these regulatory networks. And so what this plot is showing is it's showing across the x-axis I have the ratio. The y-axis is frequency, and the magenta distribution corresponds to loop transcription factors that are also in the driver position, and the cyan group corresponds to driver TFs that are also in loops. And so what this plot basically shows is just that there's a high overlap between transcription factors that are in the driver position and those that participate in transcription factor microRNA feedback loops. And so also another thing I did was I took the list of transcription factors that participated in feedback loops, and I did a simple go-term analysis. And I did a simple annotation analysis, and I basically found that transcription factors that participate in these feedback loops are associated with cancer phenotype. Misregulation of these transcription factors are implicated in a lot of cancers. And so in summary, we can combine this idea of genomic footprinting with microRNA target predictions to get a more complete view of the regulatory network of a given cell type. And within these regulatory networks, transcription factor microRNA feedback loops are abundant. And these transcription factor microRNA feedback loops occur preferentially with a subset of transcription factors. And these transcription factors that they participate in are preferentially found in the driver positions of the network. And so collectively, these results raise the possibility that microRNAs serve as a cellular buffer for these to control the expression of really important transcription factors. And so with that, I'd like to thank everybody in the lab who helped me with all of this research. Thanks everybody in the lab who helped me with this research. And with that, I'll take questions.
looked at whether there are more oncogenes or tumor suppressors to use those?
So that is actually an analysis I haven't done yet. But that would be something that would be interesting to do, an interesting and easy thing, something that I could do.
It's a really interesting analysis. It's really striking how important these interactions seem to be, given that you have so many hubs and driver transcription factors that are involved. So given that, I wonder if you've looked at functional data, so overexpression of microRNAs or knockdowns, to see whether these transcription factors can explain some of the indirect effects of the microRNAs and whether these have an impact in terms of rippling out into the network.
And that's actually an analysis that's ongoing. We're currently, I chose a subset of these transcription factors and microRNAs and that participate in these feedback loops, and we're actually doing knockdown or knockout experiments on them to see what effect they have on the rest of the network.
Okay, so we'll move on.